The DNA target is copied several times. The fragments are going to be of different size. Fluorescent nucleotides will be the end of the chain known as D-desoxy. Routinary up to 800 pair bases can be sequenced in a time. And what do you need? DNA polymerase, a primer, the 4 nucleotides, DNA template, and plus a unique ingredient that are the nucleotides D-deoxy. They don't have an hydroxyl group that doesn't allow to another strand to join. The DNA sample is going to be in a tube with the primer, the DNA polymerase, and the nucleotides. First, you gotta heat the mixture to denaturate or separate the double-strand DNA. When you get the simple strand, it cools to allow the primer to join it. Heat again to make the DNA polymerase work. This is going to add aleatory nucleotides until appears the D-deoxy. This process is going to occur several times in order to be sure of use all the D-deoxynucleotides and get random sizes. This is usually an individual DNA fragments such as bacterials, plasmids, or DNA copied in the PCR.
The main applications of the Sanger enzymatic method actually are Thermo Fisher and Illumina. The two dominant technologies are currently available. Smart sequencing depicts one type of data coordination to feed a Clinoca genomic app using SMART on FHIR, augmented with SMART on FHIR genomics. In such an app, clinical data from two systems is combined with genomics data from sequencing systems. Capillary electrophoresis is another that has been the method for analyzing selected enzymes that act over fluorescently labeled DNA substrates. It is also used for analyzing simultaneously multiple substrates, products, or reactions using different fluorescent labels. ClinicC is a flexible and user-friendly pipeline for format conversion, quality control, trimming, and filtering of raw sequencing data generated from Sanger sequencing and 3 NGS sequencing platforms, including Illumina and 454 and Ion Torrent. The last one is pyrosequencing. It requires a preparation of single-stranded DNA molecules as a starting material. The advantage of pyrosequencing is that it can be automatized to obtain hundreds of thousands of sequences in a single execution.
For the Sanger sequencing method, you will need the next ingredients and equipment: DNA polymerase, primers, the 4 DNA nucleotides and their chain D-deoxy versions labeled with different colors each, the template DNA, a capillary tube, a laser detector, computer with a chromatogram software, and the basic components for a gel electrophoresis.